And I don't think that I ever explained this, but perhaps Marie Helen did, that usually after the seminar, then the fellows have sort of a,
Yeah, I explained it.
okay. Because I will drop off and I have another meeting in an hour, but the fellows are supposed to stay and ask you questions about life, the universe, and everything. So
Okay, sounds good.
let's wait another minute or two.
Yeah. I have a video. I'm not sure if the sound will work. I wonder if I should try it while
Yeah,
we're
maybe we just try
waiting
it.
here.
Mhm.
Let's see this. Do you hear that? No, you don't hear it.
No.
I don't, but Isabel is nodding. She does.
Well, I'm not
Is she
sure,
here?
actually. I'm sorry. Maybe I hear
You're
you.
not hearing it?
Not the video, no.
So you don't hear the
No,
okay.
not the video.
No, sorry, false alert.
Not the video?
No.
Okay. Let me
But
see if
you shared
play
your
around
screen
with it.
first.
I'll I might skip the video if it doesn't work.
Okay, well, let's get started. Thank you, everybody, for joining, and hopefully we have a few more people coming on after we begin, but I'll just briefly introduce our guest today. So I'm very happy that we have Dr. Trevor Charles joining us and willing to give his time to share some of his story with us. So Dr. Charles is an accomplished and decorated scientist and educator. He's a professor at the University of Waterloo and has, a good record of all the things that professors do. But in addition to that, also has an entrepreneurial streak, and that's really the side of his story that we've asked him to speak to us about. What does it mean as a scientist to also have interests in business and entrepreneurialism, and how does that work and fit and so on. So research in Dr. Charles's lab deals with pathogen surveillance, microbiomes associated with plants and in agricultural contexts, as well as functional genomics, bioplastics, and a few other things. And in connection to those topics, he's founded several different companies. So he has Metagenome Bio Life Sciences does, well, he'll tell us more detail what they do, but some microbiome characterization services. Healthy Hydroponics offers services targeted to the hydroponics industry for pathogen surveillance and others. Earth Microbial is a company that produces inoculants, so live microbes to be applied in agricultural settings and provide benefits there to the growers. And he's also been involved in Liftoff, which is an accelerator and incubator for Black entrepreneurs. So really an amazing breadth of things, and very excited that he's going to give us a snapshot into that, so we can also be thinking as scientists, how does my expertise and my work and maybe my future career plans, how do they intersect with industry and entrepreneurial opportunities? So I'll turn it over to you, Dr. Charles. Thanks so much.
Yeah. Okay. Thanks, Matt. Thanks for the introduction. If my signal is unsteady, please let me know. I just got a notification that my Wi-Fi wasn't too strong. But yeah, I'm pleased to be here. I think that the timing is good because I gave a similar talk to my Department of Biology at University of Waterloo. And it's interesting because University of Waterloo is well known for its entrepreneurship, but that doesn't necessarily span the whole institution. So there are some parts that are very entrepreneurial. My department, the Department of Biology, is not really that entrepreneurial, but there are so many opportunities for biology and the life sciences, as I have in my title, that I'd really like to evangelize this, because I've been bitten by the entrepreneurship bug starting about 10 years ago. And there's so many things that you can do with science and business and entrepreneurship. So I just like to get the word out. So thank you for this opportunity. So there are a few companies that we formed, and they're all sort of interrelated. But I wanted to start with one that isn't on this list, and that is Metacycler Bioinnovations. And so when I gave this talk a couple of weeks ago to my department, what I did is I had one of the founders of Metacycler come up and give a short pitch. I think it was a 3-minute pitch. And I had wanted to do this, but Zoom is not cooperating. But this is a pitch from a few years ago. And so here we have, you can't hear the audio, I don't think. But this is Eugenia, one of the founders. And what I'll do is I'll just skip through some of the slides from a more recent pitch from Metacycler, just so to give you an idea of some of the things that the students can do. So Metacycler was formed by 3 PhD students in my lab, and really it's a good illustration of the types of things that you can do, because as they're doing their PhD, 2 of them were able to get accepted to a part-time MBET program. So MBET is the University of Waterloo's equivalent of an MBA. So it's within the Faculty of Engineering. It's the Master of Business, Entrepreneurship, and Technology. And one of the things that they do as part of their program, either as a full-time or a part-time, is that they build a business. And so they are able to bring this business that they've already founded to the MBET program and use it really as a case study as they're going through and as they're doing their PhD as well. So this isn't the only program that's available. A lot of institutions have these types of things, but I'm most familiar with this. So I'll try and do justice to the pitch as we go through these slides, but really, the problem that is identified here is plastic pollution. So plastic pollution affects us all. We're all familiar with that. How do we find solutions to that? The solution that Metacycler has come up with is to use biodegradable plastics, so to produce biodegradable plastics. Now, one of the big challenges with the production of biodegradable plastics, such as polyhydroxyalkanoids or PHA, is that the cost of production is not competitive with traditional fossil fuel derived plastics. And the biggest reason for that is the cost of the feedstock that's used to produce the biomass from which the bioplastic is extracted from. So there are examples of using sugar from corn, for example, or oil from canola. But wouldn't it be great if you could identify a waste source that you could use and basically upcycle so that your cost of production is reduced substantially, because that waste you actually get paid to take it away rather than paying yourself to obtain it. So what was identified here was dairy waste, which is quite abundant, and a lot of it is actually landfilled. So what can't go to to animal feed. A key part of this is the technology. So we have proprietary bacteria that are able to take that dairy waste, which is high in lactose, and convert it to a number of different PHA bioplastics. There's a number of advantages here. And we even have advantages in our system compared to a number of other PHA bioplastic producers, as you can see here. Part of it is the wide variety of PHA bioplastics that can be prepared using our method, but also the production cost is estimated to be quite low. Of course, the biodegradable plastics market is growing rapidly worldwide. And we started with technology from the lab, right? So we were working for many years developing this in the lab, eventually got some students interested in commercializing it, and they're now optimizing the product development. They're doing some pilot scale-ups, going to release their first products in Ontario, and then Quebec, and then expansion to the US. So that's the sort of the idea here. I'm sure I haven't done justice to that pitch. Eugenia is really the expert at giving pitches, and they've been able to win a number of pitch competitions, starting with that $5,000 pitch competition at on one of the first slides there, and they've gone as far away as Thailand to enter and participate in and win pitch competitions. As part of this, we have a business team. We also have a technical team. One of the things that I've noticed is that it's much easier to teach a scientist about business than it is to teach a business person about science. And this is something that I always say, because I think there's huge opportunity for scientists to get involved in business. Our technical team you see here, one of the things I want to make sure that you're aware of is that University of Waterloo has the largest and most extensive co-op program in the world. So over 70% of students are co-op students. They spend up to 2 years in the workplace by the time they have finished their degree program. So we've hired 2 co-op students this term, and we almost always have a co-op student working with Metacycler. And then our advisory team, Tiz Mekinen is a professor in chemical engineering, and I'm a microbiologist in the Department of Biology. And for us, that dialogue between biology and chemical engineering is highly productive. So that's Metacycler. I wanted to tell you a little bit about how the company was actually launched. So at the University of Waterloo, it's a little bit different than other universities. So we have something called a creator-owned IP policy. And I think it's really the only one in the world like this, although there are other institutions that say they have a creator-owned IP policy. When you dig into it, you find that there's some bottlenecks. So what creator-owned IP, the Waterloo IP policy means is that the default is that the inventor or the scientist or the engineer, they own that IP, no questions asked, and they can take it and do whatever it is that they want with it. There is sort of an obligation for faculty members at least, to report that in their annual report. So basically you just have to disclose if you have a patent application or something like that. But other than that, you're free to do whatever you want. You could license it to a company, you could take it and commercialize it yourself, anything you want. But that doesn't mean that we don't have a tech transfer office. So we have something called WatCo. And the reason why I'm mentioning this is because this was working with WatCo was how Metacycler decided to launch their business. So they pitched it to the commercialization office, to WatCo, and WatCo liked what they heard, and they actually paid for the first patent application. And they also paid for the incorporation and the legal fees and that sort of thing in return for a small amount of equity. I think it was less than 5%. So although there's an open IP policy, there is an option to pitch it to the commercialization office if you would like some assistance. And here, the benefit was having the patent paid for at the early stage. Okay, so I want to go through a little bit of science, and I'm going to circle back to some of the other companies that we do that are sort of more of an agricultural focus. But in reality, because this involves food waste and also production of biodegradable plastics, it is sort of considered as an agritech application and commercialization. So these are the polyhydroxyalkanoids. They're produced naturally by the bacteria. Many bacteria will produce them. Those, what you see inside here, I'm not sure if you can see my cursor, but those are, actually, maybe I can laser pointer here. Those are essentially stored as we would store fat, right? So the carbon and energy reserve for the cell. And they typically will accumulate that when there's an imbalance. So there's a lot of carbon available in the nutrient compared to some other limiting nutrients. So it could be nitrogen, could be phosphorus, it could be oxygen, number of different things. So the simplest one is polybeta hydroxybutyrate. That's a 4 carbon, and 4 and 5 carbon are considered to be short chain length. So here we have hydroxyvalerate, and then above 5 carbons, so 6 to 10, those are medium chain length. And sometimes I think we can have up to 12 carbons. And the physical properties will vary according to the polymer composition. So the longer chain length you have, the greater the elasticity and the lower the tensile strength. And you can combine these different copolymers within a single polymer to make a variety of different characteristics. So this is just showing a matrix here with Young's modulus and elongation at break. Those are 2 important characteristics. And we've got a number of petroleum-based polymers here, like PVC, LDPE, PET. So that's what you have in your pop bottles, polypropylene. And you can see that basically by modifying the copolymer composition, you can get different physical properties. And the idea would be to try and match some of those existing plastics so that they can be drop-in replacements for them. Now, in order to do that, we need to do a little bit of genome engineering. So the metabolism of the production is essentially you've got the carbon feedstock entering here, goes through fatty acid biosynthesis that provides then the substrate for the PHA synthesis. Fatty acids can come in as well, and you get your polymer. So the copolymer composition is really going to be dependent on what substrates are available for the PHA synthase enzyme, but it's also determined somewhat by the substrate specificity of the enzyme. Now, we work in Pseudomonas almost exclusively, so Pseudomonas putida or alloputida, and most Pseudomonas strains cannot use lactose or galactose as a sole carbon source. So one of our first tasks for genome engineering was essentially to construct lactose and galactose metabolic pathways. And this was done by Arantxa, who's one of the founders of Metacycler, a PhD candidate in the lab. And so we just dropped this in. So lac operon from E. coli, galactose operon from a Pseudomonas strain that we happen to have in the lab. This is actually one of the plant growth promoting strains, which we had the sequence of, and we found that it had galactose metabolism. And so we dropped it in there. That's all driven by a single promoter here. And it actually works quite well to utilize that lactose and galactose. And then we also will engineer this PHA synthesis pathway, which is in the genome there. We get rid of the PHA depolymerase so that we can have a higher production. So that means that it's not depolymerized as part of the cycle, and it's just accumulated to higher levels. And typically there's these Pseudomonas have two PHA synthase genes that make the enzyme that polymerizes. Oops. Why is it not advancing here? Oh. Okay, so just going back a bit, well, we want to be able to change the substrate specificity of this polymerase here. So what we did is we did some functional metagenomics. So we had developed functional metagenomics capability for a number of other purposes. And so basically what this is mining the diversity of the genetic diversity that we have in Canada. And a lot of soils were sampled from across the country. A lot of this was done in collaboration. A number of ecologists who are working in some of these remote areas and some not so remote areas. They shipped them to us. We isolated the DNA, and then we made metagenomic libraries. And you can see here's a number of them. So essentially what this involves is you just take the DNA, very high molecular weight, you shear that into large pieces, and then you clone those pieces into a vector. And so this is a cosmid vector here, which replicates in a number of different bacteria. Our best library is almost 9 million clones, and these are all very large fragments. And this is a great resource that we can then use to isolate novel genetic material. So we isolated using a screen that I won't go into here, a number of novel clones that had genes that would direct the PHA synthesis. And I think we've got a little bit of problem here with the formatting, but we were able to essentially, by introducing novel PHA synthases, direct different copolymer composition. And we're incorporating that into production of our final product. Okay, so now let's move on to some of the other commercialization that we're doing. So these are our companies. Metagenome Biolife Science is the core company. All of our companies are still fairly small, but they're all under common ownership, and we split them up so that we could focus a little bit rather than having everything in the same company, because otherwise we get a little bit too distracted. So Metagenome Biolife Science is essentially a lab that we have off campus that provides microbiome analysis by DNA sequencing, initially to academic clients, but now we've rolled that out to a number of industry clients. We also have a DNA extraction kit. Works really well for fungi. If any of you are interested, it's called the SOX DNA extraction kit. So a few years ago, after some initial R&D that we did that was funded by the Ontario Ministry of Agriculture to really characterize the microbiome of hydroponic systems like vegetable greenhouses. We launched this company, Healthy Hydroponics Initech, that does pathogen surveillance in hydroponic systems. And then we've got Earth Microbial, which has a branch both in Canada and also in the US. And basically that develops and sells biostimulants and biofertilizers based on bacterial strains. We've had a number of people over the years, and we always like to couch this within the UN Sustainable Development Goals, which are a great touch point, I think, for anybody trying to build a business that is sustainable. You're probably all aware of the 17 Sustainable Development Goals. And we, whenever we're doing something in terms of building the business, we always like to come back and take a look at, what is the ultimate impact of whatever we're building? So the UN SDGs, they're really are surrounded at, they're really surround the biggest impact, which is eradicating poverty, which is really the greatest global challenge. And as I've indicated here, sustainable development isn't possible without eradication of poverty. We really need sustainable development to heal the planet, and you have to balance the economic, social, environmental. You can't build an effective business unless it's going to generate revenue and provide high quality products and services that people want to buy. And that whole successful system is what's going to allow us to meet our sustainable development goals. Now, interestingly, Kitchener, where I live, just beside Waterloo, is one of the leaders in the promotion of the UN's SDGs. And so this is a place called the SDG Idea Factory, which is a place for entrepreneurship and community building in the heart of Kitchener. And this is where we developed our Liftoff Black Entrepreneurship Program. Over 3 years, we had about 150 Black entrepreneurs, mostly from Southwestern Ontario, who went through the program and build their business. And it was really sector agnostic, so not just tech businesses, not just mom and pop shops, but everything in between. There was a little bit of agri-science in there. There's some food businesses as well. And I think we had a good impact on the community. And we've moved from that to the LBIH or Liftoff Black Innovation Hub that is really moving us to the next level, which also includes developing a fund that can invest in some of these companies that we're bringing along. Okay, so the UN's Sustainable Development Goals, that fits well into our vision of a circular bioeconomy. So anything that we do in my research lab or in the businesses that we build, in addition to being aligned with the UN SDGs, we also like to fit within a concept of circular bioeconomy. So this is our concept of circular bioeconomy, which includes the bioplastic work that I indicated, and also food waste that can be converted to a substrate by anaerobic digestion or leach bed reactors. That gives you a leachate that can go directly into fermentation. This is leachate that's high in volatile fatty acids that can be metabolized directly by the Pseudomonas strains that produces your bioplastics. Then they can be incorporated into product. And then we always like to avoid having anything going to landfill. So we have this concept that Metacycler is actually built around, that end-of-life product could be metabolically recycled back into plastic. And we also like to do this with fossil fuel derived plastics as well, once we solve the technical challenge of degrading that plastic and using that plastic as a feedstock for the production of bioplastic. In this example here, that leach bed reactor is also producing biogas and phosphate and nitrogen fertilizer, which can feed into a greenhouse. That greenhouse produces food that goes to food services, and then the cycle continues. So that's our concept there. And each of our companies fits into here, into that. So we've got our company Earth Microbial here. We've got Metagenome Bio, and we've got Healthy Hydroponics. One of the things that we learned and we experienced is that although you might have the greatest plans for building your business a certain way, you always have to be on the lookout for opportunity. And this is where this NTurf came about. So early on, we were able to do some turf trials at UC Riverside. So this is trials where our products were competed alongside more traditional chemical fertilizers and pesticides. And there's a particular combination that performed really, really well on turf. So we took those and we commercialized it immediately, and that's where a lot of the traction is coming in the market. And so we're basically got a sales team that spends the day, a very small sales team of only 2, but very effective, where they spend the day cold calling golf course superintendents, setting up meetings and then making sales. And then based on that, we generate revenue, and then that allows us to build the business. So a turning point for us, the original company was started in 2014. A turning point came from us moving off of campus to this space here. This used to be a machine shop. We leased some space here. And this is what it looked like before we did a makeover. And this is what it looks like now. So it's still not like you would find in an academic research lab. We use, for example, kitchen countertops that we have over here. We had some friends help us to build that out, the supports for it. And you know, a lot of it is makeshift, but it's very effective. Our inoculant production is in the back here, and then this part here is all for our molecular work. So there's our production of our products. We're still doing it pretty small scale. It's still flask-based, but that's the most effective production method that we currently have. And we're just about to embark on scaling that up to bioreactor production. So that's the outside there. And now I wanted to move into basically the agricultural applications of some of the products. So this is a really important part of the world that's centered here. This is one of the southernmost parts of Canada. So this area here. And that is actually below the it's further south than the northern California border. Okay, just to put it in perspective there. So this is Leamington, near Windsor and Detroit, which is over here. And this is where about, I would say, 1/3 of all vegetables in Canada are produced. More than 50% of the greenhouse grown vegetables. And I mean, the technology that has developed in this area for greenhouse grown production is really nothing short of amazing, and it's really world leading. I think the efficiency of production is even greater than what you find in the Netherlands. So you can see those greenhouses from space, or from satellite, I guess. But this is another example. This is in Spain. And the technology of the greenhouses is not as advanced here, but the fact that you can see such an impact of that what is mostly plastic greenhouses is really nothing short of amazing. So half of the vegetables produced in Canada are produced in greenhouses through hydroponic production. This is a rock wool substrate here. This would be a typical commercial production system, although there's many, many different ones. But the key thing is that they're not grown in soil, and the fertigation systems will deliver the nutrients that are, fine-tuned for optimum production. Many different types here. So we've got greenhouses, which are sort of more typical, but we've also got shipping container farms, vertical farms, aquaponics that incorporates fish and other types of animals like shrimp and that sort of thing. A key thing to make note of is these shipping containers and many of the vertical farms don't use natural light. They will use LED light. Many advantages: local, sustainable, adaptable in scale, stable year-round production, reduced footprint, and also addresses food insecurity in remote and urban communities. And also, I think, especially in our current trade environment, being able to be self-sufficient on basically on a national scale is really, really important. So these produce half of the Canadian vegetables with only about 1% of the acreage. So here's our Healthy Hydroponics project. That was funded by the Canadian Agricultural Partnership and also NRC IRAP. And basically, our premise coming into this is that those hydroponic systems which don't use soil don't really pay much attention to the microbiome unless pathogens are considered. And we always think about how important healthy soil is to good crop production. But in this case, there's not really any soil microbial community to speak of. So we would really like to have some insight into the composition of the hydroponic microbiome so that we can optimize it for production. These systems we call controlled environment agriculture, or CEA. So maybe I'll skip through this quickly. But one of the key things here I would highlight is that there are many examples, many instances of growers losing hundreds of thousands of dollars due to crop failures because of pathogen outbreaks. And when you think that, for some of these crops, they could be in the greenhouse for the better part of a year with 9 or 10 months of production from the same plants. And if you have to cut your growing season short, that's a huge impact on your business. So the idea here is that we can, using genomics, so sequencing and qPCR, get a really great overview of what the microbiome is. When we first started this, really as microbiologists, our idea was, well, how can we get an idea, a good view of what the microbiome is, and then improve it? But when we looked at the business opportunity was really with the growers wanting to know whether or not they had we're at risk of pathogen outbreak, right? So disease outbreak due to pathogens. And so by developing a surveillance program, you can get some early warning so that you know whether or not you are really at risk of having an outbreak. So we have a fast turnaround time and also much higher specificity than a lot of other alternatives. And also we have a proprietary pathogen DNA database that allows us to classify the pathogens with high accuracy. Okay, so skip through this. We also see that in addition to reduction of disease outbreaks, there's also potential for improvement of the efficiency of these systems. So by improving the microbiome, could we potentially reduce the light requirements for production, also heating and cooling, reduce fertilizers, reduce disease, and at the same time increase yield and increase product quality. Part of this is through the ability to provide some flexibility in the inputs and the conditions. So for example, if we are able to improve the microbiome so that the plants can still produce under a greater range of EC or pH or salinity, then that really helps this overall equation here. So Healthy Hydroponics provides microbiome monitoring for crop health and food safety. That's through high throughput sequencing. Essentially, the grower will filter out some nutrient solution from their system and send us the filter. We then will extract the DNA from that and carry out our analysis. Our initial project had 13 farm microbiome profiles that were developed. And as you get more and more data, then that database has increasing value. And of course, we use machine learning in order to do all of this, as we do for all of microbial ecology. So in that initial study, we were able to find out that there are a number of characteristics of the microbiome. So the first is the different types of organisms that were present. So there's a lot of Pseudomonas, and this is the different crops are indicated by these different shapes and colors here. The Pseudomonas was really widely distributed, and it was really broad variety of different types of Pseudomonas that was present in these different systems. And they're often dominate, so they can be over half of the microbial community. We found some interesting organisms that often you don't find in soil, but you find in aquatic environments. So these Oceanospirales. There was one of these called Pocalibacter that we actually isolated and is a potential product as an inoculant product. But these probably are due to the fact that it is more of an aquatic environment than a soil-based environment. And in some cases, the salinity can increase, and that's why you might have some of these that are more salt tolerant. So this Pocalibacter species is known for its salt tolerance. Now, one of the types of bacteria that you often find in soil are bacillus, right? Soils are well known to be in bacillus. And interestingly, the only bacillus that we found, and it was pretty low abundance, and it was not in all systems, were these two clades here. And it turns out that these were products that were actually added to the system, right? So these are bacillus inoculant products that were added. Otherwise, you don't see bacillus in these systems. So it's just sort of a snapshot or an indication of the types of things that we find here. And I'm sorry, I should have also talked to you about some of the fungal microbiome that we see in there, but I had taken out those slides and didn't occur to me until just now that I should have kept them in. Here is our growers portal that I just give you an indication of what the grower sees in terms of their data and how they're able to follow that over time. And then, of course, in order to be a real business here, you have to have products and services that people will pay for. Okay. Trade shows are also really important to get the word out. Here we are, probably this is a Canadian Greenhouse Conference or someplace like that. And this is really great for meeting growers and also potential collaborating businesses, right? And this is an example here. We partnered with SGS, which is a multinational lab services company, and they offer our services to their clients through us, and we're hoping that is a fruitful partnership. So they're white labeling our services, and we're also developing new tests with them. I think I've got a couple of minutes just to talk to you a little bit about Earth Microbial. So Earth Microbial is our business that develops the microbial inoculants. And you're all microbiologists, you know how important the soil microbiome is. Basically what we do is we've got a culture collection that we've developed over time. We screen that culture collection by DNA sequencing, and also we test them on plants. There are certain traits that we're particularly interested in. In some cases, they are isolated through specific types of enrichment that enrich for some of those traits. And then they will go to production, government registration, then the final product. Government registration, so most of all of ours are registered through CFIA, Canadian Food Inspection Agency. That's a process that's a multi-year process. One of the interesting things is that registration is different in different jurisdictions. So in Canada, you know, the whole country is covered by Canadian Food Inspection Agency. So you just need to do your registration once, but it can take you several years from the time you start collecting data to the time you submit it, and then finally get your authorization. And that can be a real impediment to business. In contrast, in many of the US states, or in the United States, it's the individual US states that you register through for the types of products that we're working with, the biostimulants. And so you have to target which states you want to sell in, and then they all have different regulations, they all have different requirements, and so you have to deal with that. Here are some of our products. We've got NGuard and Fix. Our first product was N-Stim. It disrupts the production of ethylene, stress ethylene. So it's particularly good at transplant for some of the vegetables into these greenhouse systems. We have NGuard, which increases tolerance to pathogens, and Fix, which enhances nitrogen fixation. And then N-Turf is a combination of the N-Fix and the NGuard. And that's the one that we sell into the turf market, the golf courses. Oh, yeah, that's what I was going to say. The reason we call them N is because they're all endophytes, right? So they all will infect the roots of the plant, and that's where they set up residence and provide their beneficial activity. ACC deaminase is a trait for N-Stim. It reduces the stress response through this, the ability to use ACC as, which is the immediate precursor to ethylene, as a nitrogen source, nitrogen and carbon source. So it will degrade that into ammonia and alpha ketobutyrate, which can then be taken up by the plant and by the bacteria, and that's the benefit to the bacteria, and in so doing, it reduces the stress ethylene. So that's the basis of our N-Stim product. Interestingly, that reduction in stress can, and this is a commercial trial, one of the first commercial trials that we did in a tomato greenhouse. We got almost 20% yield increase. And this is the final product yield increase. And this is almost, the growers tell us, you shouldn't tell people it's almost 20% because nobody will believe you. That even, a 2% or a 3% is almost unheard of. But the thing is that that 20% increase is relatively reproducible, whether we do it in the growth chamber or we do it in the field or we do it in the commercial greenhouse. We always get around 15 to 20%. Here's some examples of our turf grass trials. This was done at UC Riverside. Some of this was done in our lab at University of Waterloo. And it's really taken off, this particular product. Sales are really going to the point where it's hard to keep up in terms of our production. We're looking at many different crops, so we're looking at different trials that we're doing, but really at the end of the day, it's about the ROI for the farmer, for the grower. Does it actually make sense for them to use our product if it's not going to provide some benefit in the end? And so we also have some products that are in development for the next year, including for flowers, for desert soils, for soybean treatment, for leafy greens, and for strawberries. And so I think that's the end of my presentation. So, of course, just like any type of research, you need different types of support in order to develop for the business. So we've been happy to tap into a number of them here. So OMAFRA, Ontario Ministry of Agriculture, Ontario Agri-Food Technologies, NRC, NSERC, BioEnterprise, Weston Family Foundation, MyTax, BioTalent, and Canadian Agricultural Partnership. And of course, we're always looking for new sources of funding to develop lots of interesting things. So maybe I'll stop there. And this is my lab from campus. Oh, actually, do I have a few minutes? I was supposed to talk about the entrepreneurial environment in Waterloo. So I think I have a couple of minutes that I'll spend to do that. So the University of Waterloo has Velocity Incubator, and anybody, whether you're student, staff, or faculty, can apply to join Velocity and build a business. And you get support in terms of coaching and mentoring, and even some space. So here's an example in the older facility, and this is the Innovation Arena, which is just opened up across the street from the original Velocity lab. And so this is 90,000 square feet in downtown Kitchener. I think it's one of the largest entrepreneurship innovation spaces in North America. And so Velocity has the top floor, and then the bottom floor is for a number of organizations and companies. But it's not only tech-based entrepreneurship, but we also have greenhouse, which is it's associated with United College, which is one of the affiliated colleges at the university, and its focus really is on social impact entrepreneurship. So this is in many cases nonprofit, but some for-profit as well. And here, really the goal is social innovation, right? And they have a new social innovation minor that you can take along with your regular degree. Flint Hub is the incubator for Indigenous startups, and that's coming along quite nicely on campus. We also have the Grable Peace Incubator. This is part of the Peace and Conflict Studies at Conrad Grable University College. And then I mentioned the MBET program. They also have something called the Entrepreneurial PhD Fellowship at the Conrad School of Entrepreneurship and Business, where you build a business along with doing your PhD. And then another thing that's really, really interesting, and maybe I'm a little bit biased because my daughter went through this program a number of years ago, but the Stratford School of Interaction and Design is really interaction design and business, is really a business program that is focused on tech, but is within the Faculty of Arts, and is meant for individuals who aren't necessarily building a career that's based on technical ability, but is based on all of that other ancillary skills that are required for tech businesses and other businesses to be successful. There's other important ecosystem players. So we have the Accelerator Center, which is just on our North Campus, which runs a number of programs and also provides incubation space for businesses. And then Communitech is for specifically tech-based businesses. Just skip through there. And then also, this all ties in with the focus of the University of Waterloo, which is really the global futures. So we have societal futures, health futures, sustainable futures, technological futures, and economic futures. So everything that we do in entrepreneurship needs to tie in with all of these futures that sort of guide what we're developing at the University of Waterloo. So that brings me to the end, and thank you very much for your attention.
Thanks for that, Trevor. It was very interesting. So we can, if there's a non-fellow who wants to ask a question before we sort of turn it over to the program fellows, we could do that for a few minutes. I don't know who we have. Isabel, anything you want to ask?
Yes. First, I want to thank you very much. It was a very interesting talk, very interesting. And so I'm Isabel Benoit-Galber. I work at Concordia University in the Genomics Center of Concordia, and I work with fungi. And so I have a bit of a culture of science related to company. I spent 8 years in the Netherlands before coming to Canada. So when you mentioned the greenhouses, I was excited. So I had a question. I have many questions, but I will just focus maybe on the first part of your talk. And you mentioned the waste that you are working on, including plastic. And I was wondering, I've been told many times that in order to address the waste issue, the problem is separation of the waste, right? So when you you have to, there are so many things in there that you need to target. You mentioned lactose degradation, but there are so many other things. So how do you do with the separation of waste? Is there something that you had to address, or how did you cope with that?
So that's actually a really, really great example because there's so many waste products, but there are some waste products that are sort of the low-hanging fruit, right? That you can address more readily. So what we identified was the milk permeate, which is high in lactose. It's got some other things in there as well. But essentially, we can use that directly. So we don't have to separate it at all. So we just add it to the growth media, and then that provides the carbon source as the lactose, and it's probably using some of the other nutrients in there as well. We haven't done a complete analysis just to see how those are utilized and incorporated, but it works surprisingly well. So that's an example where we don't really have to deal with that separation of the waste because we use the waste directly.
Right. Okay. So do you have in mind any other example where you have to separate? Because I mean, there are many, what I mean is that this is somehow, and I don't know if I can say kind of an easy waste because you don't have to separate.
Yep.
But how do we address then, and maybe it's a question we cannot answer here, but how do we address all the other waste? Because plastic, for instance, we separate plastic, but it's still, mixed with a lot of things. And plastic itself, it's so many different types of plastics. And then if you address in the field, in the waste field, it's also very complicated, right?
Yeah. Well, so for food waste, so just general food, like post-consumer food waste, we're working with some civil engineers who have these anaerobic digesters or leach bed reactors. So in that case, we're transforming the waste into a leachate that can feed directly into the process. So there isn't really that separation.
Very good.
Actually, so I just noticed, so I see Kelsey on the screen. Kelsey is actually doing an internship at our metagenome and Earth microbial. And so you can see in the background there, I know exactly where you're sitting.
Thank you very much. I have other questions, but I don't want to monopolize
Yeah, no problem.
Yeah, I forgot to mention that Kelsey's doing her EvoFunPath internship with these companies. So thanks for filling that in for me. I think it's time that we turned over to the fellows. Now, I don't know whether any of the fellows have signed up to lead the question and answer period. Has anybody?
Yeah,
anybody?
some registered. We have Mayara and Mariana.
Oh, perfect. Okay.
Yeah.
So in that case, I'm going to sign off and the other non-fellows can sign off. And I hope, I anticipate questions about sort of general career strategy and just, may not be so scientific, Charles, but whatever, or Dr. Charles, but whatever you're willing to entertain for questions would be appreciated. And let's just thank the speaker before we make that transition. So really grateful. Thank you. Okay. And I'm going to leave, but.
Bye. And I'm going to stop the recording, guys, so you can ask your question. So do you want to take a few minutes, or are you good to go? Dr. Charles, do you need a little break, water or something? Or you're fine? Guys, are you willing?
No,
Can we
no,
go right
Yep.
away? Okay. So I'll let you, Mayara and Mariana, I'll let you guys handle it.
Okay, thank you. Can you just remind me how long is the Q&A?